Welcome, my dear students and others, to this Chapter 14 coverage of digestion, in which we will talk about breaking down lipids. This contrasts with breaking down proteins and breaking down carbohydrates, which we discussed in our previous two videos, linked to in the description below. So, as we discussed way back in Chapter 11, lipids contain more energy than proteins and carbohydrates. Why? The reason is because lipids are composed of thermodynamically more energy-rich carbon-carbon and carbon-hydrogen bonds, which give off more energy during combustion than the numerous lower-energy carbon-oxygen and carbon-nitrogen bonds found in proteins and carbohydrates. Unfortunately, this presence of carbon-hydrogen bonds in lipids creates one problem. They are very non-polar, which means they do not readily dissolve in the aqueous environment in our bodies. So how in the world can our bodies transport and digest them? I'm glad you asked. So, lipids are ground and mixed into the stomach to become something called an emulsion, which kind of looks like a mixture of oil droplets sitting in water, as you can see in this picture of oil and water not really mixing. Anyway, after these lipids leave the stomach, bile salts from the gallbladder, if you happen to be one of those people who has a gallbladder, unlike me, I don't have one, I'm a gallbladderless freak, had to have it removed. Anyway, bile salts from your gallbladder insert into the middle of the oily liquid droplets, which helps make them a little bit more water soluble, since bile salts are amphipathic, which means that they are partially soluble in both polar and non-polar environments. For interest's sake only, I wanted to point out that glycocholate, an important bile salt, looks like this. Take a moment and look at that structure. So with that in your mind, I think you might recognize that glycocholate is structurally very similar to cholesterol, which looks like this. Why in the world did I show this to you? Well, as it turns out, unsurprisingly, glycocholate, this important bile salt that helps digest lipids, is synthesized by our bodies from cholesterol, which we obtain from our diets. This synthesis of glycocholate from cholesterol happens in our livers. All right, so once lipids are made more soluble by bile acids in the upper intestine, pancreatic enzymes called lipases break apart the triacylglycerols through the following reaction sequence. H2O comes into one of the sections of triacylglycerol, forms a bond between its oxygen and this carbon, pushes electrons up onto this oxygen, then slaps it back down and kicks these electrons down into this oxygen, thereby severing this oxygen carbon bond right here and releasing the free fatty acid with an oxygen on it that came originally from the H2O. That, of course, gets deprotonated at physiological pH, so you have a free fatty acid, and you've effectively shaved off one of these acyl groups off of the triacylglycerol, which simplifies it down to diacylglycerol. Lipase enzymes do this again. They shave off another one of these acyl groups to reduce this down to a monoacylglycerol and release another free fatty acid. Okay, so with all of that said, I want to remind you that lipids, the term lipids, actually refers to 5 different subclasses of molecules: free fatty acids, triacylglycerols, phospholipids, glycolipids, and steroids, as we discussed in an earlier chapter. Triacylglycerols, shown right here, and free fatty acids that are shaved off of the triacylglycerols are only 2 of these different types of molecules or lipids of the 5 total lipid classes. So once triacylglycerols have been broken down into individual free fatty acids and monoacylglycerols, they now get carried as micelles into the intestinal epithelium, where they are then absorbed through the plasma membrane. Now, the micelles mentioned here, by the way, form spontaneously as the fatty acids and monoacylglycerols point their polar heads toward the aqueous media in our intestines, kind of shown here in this simple model as pink polar heads, and their non-polar tails inward, as depicted in this figure. Very similar to phospholipids in the phospholipid membrane, by the way. All right, so here's the amazing part. Once the micelles get shuttled back into our intestinal cells, they then get converted back into triacylglycerols and get stored in lipoprotein transport particles called chylomicrons. The chylomicrons are ultimately released through the intestinal wall into the blood, where they can be picked up and used by other cells all over our bodies. So after eating a fatty meal, our blood appears milky because of the high content of chylomicrons. Incidentally, bile salts assist in helping fatty acids and monoacylglycerols to form micelles. People who have liver disease and cannot make enough bile salts will secrete large amounts of undigested fats in their feces, a condition known as steatorrhea, after the common fatty acid stearic acid. Whew! So the ability to maintain sufficient, but not excessive, energy stores in our bodies is called caloric homeostasis, or sometimes energy homeostasis. Because many people take in more calories than they burn and our bodies store the excess, obesity has become a near epidemic across our country, where nearly 30% of all adults are obese, according to our text. So how in the world do our bodies know when we have eaten enough and can stop? Well, once we are full, short-term signals relay feelings of satiety, which is fullness, from the gut to various regions of the brain. This occurs through signal transduction pathways that involve a G protein-coupled receptor that is bound by CCK, which we mentioned earlier in this chapter. Another signaling molecule is glucagon-like peptide 1, or GLP-1. Now, just so you know, signal transduction pathways and G protein signaling were covered earlier in Chapter 13, which we skipped in our class, but you are welcome to review if you want. I won't test you on it, but you can read it if you're interested. In essence, they function kind of like the board game Mousetrap by passing a message from one receptor to the next all the way down to the brain. And if you have no idea what Mousetrap is, or even if you do but want to, you're welcome to watch this YouTube video, which I'll post a link to in the description below and floating over my head somewhere, in which you can watch a classic old commercial for this board game. So long-term homeostasis is regulated by two hormones, leptin for fats and insulin for carbohydrates. Insulin regulation is covered on page 226 of Chapter 13, which we skipped, but you are welcome to read on your own if you wish. Again, I'm not going to test you on it, my students. This figure attempts to depict the interplay between our digestive system and our brains in order to achieve and relay that feeling of fullness or satiety. And that ends our coverage of Chapter 14. Thank you, my dear students and others, for watching these videos. I look forward to seeing you again all in class. Please have an enjoyable rest of your day.